and you have a project that involves moonlight being a certain way, and you were planning on doing your work Thursday night, you should not plan on doing your work Thursday night, because Thursday night it's going to be cloudy and rainy. So you should be checking the weather as your work approaches, because if you stick with the plan that you had before, you might not be able to do it this week. And if you don't do it this week, you might not get to do it at all, because plants will have senesced, the flowers that you were counting on being there are no longer going to be there, and things like this. So as you approach lab tomorrow especially, but also on Thursday, we have practically all of the materials put together for all of the groups at this point. I can only think of a couple of small things that we don't have together. You, in theory, can start working on your field work like now or tomorrow, because Wednesday, Thursday, Friday, Saturday, and Sunday, it's supposed to rain. And not little rain, but like if you look at Weather Underground, it's just rain. So keep that in mind as you think about what today and tomorrow hold, so that you don't just get negatively affected by weather. And
I do have your plan feedback from Professor Nickel to hand back to you today. So if you didn't stop by her office to pick it up last week, we need to get it from you today.
So that's the only thing about labs, so just think about that. The other thing is there was no preparation homework for what I'm going to cover today, because I'm not sure how far Dr. Reynolds is going to get into class. So there is preparation homework. I will post it so that you can have what would have been given as preparation homework, but I'm not going to ask you to turn it in. And there will be practice homework over the stuff that I'll start covering today. But I don't know if I'm going to assign that to be due next time or the time after that. It'll just depend on time.
Depends on how much we get through today. So we'll try to be really clear about those. Okay, so today we're going to talk about a couple more of the quantitative genetics and evolution topics that we started on Friday. We are going to discuss Fisher's fundamental theorem. We're going to talk about how that theorem can be used to project the evolution of traits over generations. So if you know some information about the heritability of that trait, and the selection on that trait, can you predict what will happen within one generation, 2 generations, 3 generations? And then we're going to talk about heritability and how it may change in response to selection. Before we get to those, I just want to give us a brief reminder. I'm going to go through these slides pretty fast that are the reminder part, okay? Because they're a reminder. They're not new stuff. I know it might not feel super comfortable yet, and that's okay. I do expect to see you guys in my office to come talk to me about this stuff before the exam, though, because if you don't, you're going to be sad when you're taking that exam. And maybe even sadder when you get it back from me. So none of you, I don't think, have come to visit me in office hours yet about this class. And so that's something you should start to just add into your schedule for some of these things as we go along. Because starting kind of now, we're going to start talking about more and more kind of complex stuff. So certainly before the next exam, come in and talk to me about quantitative genetics in response to selection and make sure that you just got it down, okay? All right, so remember we talked about genetic variation for quantitative traits. Another word for quantitative traits is polygenic, right? Poly meaning many, genic referring to the number of genes, right? Okay, and we talked about the Hardy-Weinberg equilibrium. This is just that same slide from last time. One trait determined by many genes. Each gene adds to the phenotype, and then how much each gene adds to the phenotype is determined by those specific alleles of that gene. Even though it's called a quantitative trait, it doesn't have to be continuous like height. Height is a quantitative trait. It has continuous measure, right? You're not either short or tall. You can be sort of anything in between. You can have a quantitative trait that is discrete, either having heart disease or not, right? You got little things, environment, genes, etc., adding to that whether or not you develop heart disease. But you either have it or you don't, right? So that's a discrete version of a quantitative trait. Remember we talked about genetic variation in quantitative traits. Quantitative genetics assumes that, most of the time, it assumes that all of the alleles that you're talking about, none of them act in a completely dominant way, and that all of them are somewhat additive. And I drew sort of this number line of allelic effects, right? And so you could be on one end of the number line and you could have no effect on phenotype as an allele. An allele could have no effect. And that would be like a true recessive. Most true recessives are just broken genes. Some of them are not, but most true recessives just are genes that don't work. So they don't add to the phenotype. And then you can go all the way to the maximum contribution of this gene to the phenotype. And I used silly examples of meters in height, but I think I'm going to use millimeters this time. Okay, so in this example, I've put 3 different alleles that you could potentially have. These 3 alleles exist in the population. These 3 alleles don't exist in the same person, though. Why? Why couldn't you have 3 alleles for this gene in one person?
You only have 2.
You can only have 2, right? You're diploid. You've got one copy of your gene from mom and one copy of that gene from dad. But we can have more than 2 alleles in the population. This is an example of a gene that has 3 alleles in the population. One allele adds 0 millimeters of height to your phenotype. One allele adds 2 millimeters, and one allele adds 4 millimeters. Okay? So in this example, there's those 3 possible alleles. Because it's a quantitative trait, we have lots of genes, though, adding. And a simple way, a simplification of it is just to assume, like, okay, either you're going to be, it's an allele is going to be either totally not adding anything, recessive, or it's going to add 1 unit, right? And so we said that's one way of kind of just imagining how it might be. And so we did this example with 3 genes. And we decided, okay, well, any lowercase allele is going to be totally recessive. It's going to add nothing. And every uppercase allele is going to add 1 unit to the phenotype. That's something that's not super realistic, but it helps you envision how additive alleles can impact a quantitative trait. So the genotypes are super crazy. But if you use our simplification, where lowercase adds 0 and uppercase adds 1 unit, then we have these offspring where, at least for this batch of genes, they add either no height at all or 6 millimeters to the height. All right. We talked about the Hardy-Weinberg equilibrium, and is there a significance in the fact that it equals 1? Why does it equal 1? 1 is totality, okay. So if you have 2 alleles possible for a gene in a population, if both the alleles of both frequencies are accounted for, it should equal 1. If there were 3 alleles in the population and we only accounted for P and Q, then it wouldn't add to 1, right? Because 1 equals 100%. Okay. But in this example, Hardy-Weinberg is usually given as a 2 allele system. Okay. And I wrote this out for you guys, and I'll post this just to kind of summarize what we talked about. If you have this large randomly mating population, as long as you don't have selection, migration, or mutation, then the allele frequencies should stay the same from generation to generation. So you won't get evolution, right? Because allele frequencies changing over the generations is the definition of evolution. And you can accurately predict the genotype frequencies from the allele frequencies and vice versa. Okay? So as long as these assumptions are met, then your allele frequencies and genotype frequencies should stay the same from generation to generation. But we talked about an example in which that was not the case, yes? What was the example we used? The Manx cats, right? And why do the Manx cats violate those assumptions there? Okay, so the homozygous ones are lethal, and Gabe, what you were going to say? Homozygous dominant, yes, that's true. Yeah. So which, this doesn't say anything about being homozygous dominant though, right? Which of these assumptions is it violating? Not mutation. Selection, okay? So one of those genotypes is strongly selected against, and zero fitness, right? Oh. There we go. And we gave an example of, okay, well, what if we start out with the Manx cats where each allele has a frequency of 0.5, and if we follow them over the generations, because there's selection, we don't get allele frequencies staying the same. If we start out where they're each at 0.5, they pretty rapidly change. All right, so now we're going to transition to new stuff. Do you guys have questions about the old stuff first? Okay. So we're going to talk a little bit about selection and how it interacts with quantitative genetic variation. So let's say that this is just our height phenotype again. We've got sort of arbitrarily assigned short and tall, and then we have the frequency of those phenotypes within the population. Well, if I'm feeling particularly evil, I decide short people got no reason to live. You guys know that song? Short people got no reason to live. You know that song? It's a real song. I disagree with it.
It was terrible. It was the 70s.
It was the 70s, yeah. Most things were terrible in the 70s. Okay, so I was born then. That was terrible. It was a horrible thing. Okay, so if I decide that I don't want any short individuals in my population, and I take those out, that's indicated by this sort of red blob, and I say, okay, well, I don't want these guys. Okay. Then what's going to happen to my distribution in the following generations if we assume that height is heritable? It gets skewed. Okay, so which direction would you expect it to go? Toward the tall guys, right? Okay, and so we started out with this dotted line distribution, and because I removed a bunch of these guys, and I removed a particular proportion of them, then we have selection. The force of selection is then the distance between the original mean and the new mean. Okay. So we've got selection pushing over to the right. That's directional selection, but we don't always have directional selection, right? You guys know of the Finch example. Did the Finch example have directional selection?
They had disruptive selection.
Disruptive selection. Okay, somebody else tell me about the distribution of the beak phenotypes in that population. Connor? What do you remember about the distribution of beak phenotypes in the finches?
Sure.
Did it look like just a hill, or did it look like two hills? Two hills, gotcha, okay. And what was the concept of why some finches had small beaks and some had large beaks? Yeah, so the small beaks allow you to have a very dexterous hold on these little seeds, and the large beaks help you be really strong and crack open these really big seeds. Why would you not just want a big beak? Doesn't it seem like then you could just crack open all of the seeds?
If like if the beak's too large, they may not be able to like pick up the smaller seeds, so it would be harder for them, like, so to eliminates eating smaller seeds.
Okay, might not have the dexterity involved. All right. What else can you think of as far as you got a bunch of finches on the island? You guys are all wanting all the seeds.
They're all competing for the same food.
They would all be competing for the same food. So if you can specialize, right, and have this very dexterous small beak or this strong large beak, then you might do really well. And in fact, those individuals with medium beaks are going to be competing with both the specialized small beak and the specialized large beak. Who do you think is going to have the lowest fitness in that case, given their competition? The medium beaks. Yep, Gabe got it back there. So, sorry. So in this case, the medium beak guys are getting removed, and we have disruptive selection causing two phenotype peaks, right? It's pushing them to the extremes. What if being extreme is bad? In humans, if you have too much insulin-like growth factor, or in dogs, if you have too much IGF-1, you start to have bone and musculature problems, right? Dogs can get hip dysplasia. Humans can have all kinds of problems with being too large, too tall, their bones are too long. So when you get to a certain size, you start having medical complications, right? The same as if you're too tiny, right? Teeny tiny dogs, teeny tiny humans, teeny tiny anything. Well, not teeny tiny anything. Teeny tiny mammals in general can have difficulties. So certainly for humans and dogs, it's probably more optimal to be of a moderate size, yes? And so in that case, the extremes are selected against, and you have something called stabilizing selection. So you have different kinds of selection, right? Finish what you're writing down, and then I want you to turn to a partner and talk about the types of selection and explain them. Okay?
So directional selection selects towards a certain direction, so we will like population. Disruptive selection pushes towards the two extremes, like selection,
No, they're not. I can shift it. It's not that I posted them and then I just. So what I'll do is I will post all of them, probably just in one presentation. So I'll just post them all on each class. And I should be able to do that also almost right after this. Yeah. And so in that case, Right back. And so it's always relative value here. Thinking about fitness. Yes, and this morning as I was looking at all those balls there. I have a good number. Okay, so Isaac brings up a good point. What if you have a mutation that suddenly gives you something good? I've been talking about certain phenotypes caused by alleles and genotypes that are unfavorable. What about something where you have something that is favorable? Well, that's a good point. And really what you need to just try to keep in mind is that whether something's favorable or unfavorable is always relative to the other phenotypes in the population. So the example that we talked about was the ability to digest lactose. It's totally fine if you can't digest lactose as an adult. You're probably going to be okay. But it might be better to be able to if you're in particular environments and parts of the world. And so your fitness, relative fitness, it's always relative, right? So your relative fitness is what's important in the response to selection. So in that case, that might be something where, it's going to be directional, where these guys aren't terribly impacted, certainly, but they do have lower fitness comparatively to those that can digest lactose. So it doesn't always have to be selection against something. You can think of it as selection for the other thing, right? Just sometimes it's a bit more clear to think about selection against because you can quantify which fitnesses are lowest pretty easily. Asa?
What are the different positive selection and directional selection? Positive selection and directional selection.
What's the difference between positive and directional? Positive selection would be, it's sort of like what I was just saying where you're talking about whichever phenotype is favored, and you would say positive selection for this. Negative selection is against something. And then these, directional, disruptive, stabilizing, are directions in which the phenotype will respond. And so in each case, you could talk about something being negative or positive selection against one phenotype or the other.
I usually try Isaac's example graphic by rational.
Hang on just a second. Okay, so in Isaac's example, it would be directional positive for the gain of the ability for lactose.
Yeah, we're getting into some kind of subtleties here. In order to keep it straight in my mind, I almost always think about selection against.
Mm-hmm.
Simply because if there is a new mutation and it makes one individual in the population better than all the rest, then all of a sudden, all the rest of those are somewhat less fit in comparison. And so they're not as easy, they don't survive as well or reproduce as well as the most fit individuals. So regardless of the situation I find myself in looking at population, I almost always translate that into selection against the best fit individuals.
And I think it really helps as you get further and further into evolution and you start looking at these more complex situations. It's just easy to keep things straight in your brain to think about selection against the less fit. Okay.
Are you going to mention, so thinking back to Isaac's, one of the other issues with a new mutation arising that is positive, when it arises, if it arises through mutation, it's one of them.
Right. So there's a 50-50 chance really that it will be lost in the first generation of its existence, and no one will ever know that it did exist.
So a lot of mutations, they just drift right out of the population as soon as they come into the population.
Right, and we'll talk about drift in genetics next year, certainly. Okay, so what if we didn't have as much genetics variation? What if So here's that directional selection example. What if we didn't have the alleles that gave us this part of the distribution, but we still had selection against short? How would that change this response down here?
Would just make it more of a medium? If you didn't have the taller alleles, but you had selection against the shorter alleles, would that just make it kind of balance out to more of a medium, right?
It would still be directional, but it couldn't go really tall, right? It could only go a little bit. So your response to selection, you know, you're still doing the same amount of selection, okay? But your response can't move that distribution as much to the right. Absolutely. So you would have to wait in that. So if, for some weird reason, your goal was to get, well, not weird, this happens in agriculture all the time. Let's say your goal is to get really tall broccoli or whatever, then you would really have to just wait for new mutations to arise and stay in the population that would provide you the alleles that would give you those taller individuals, and that might take a really long time, right? In fact, a lot of people will go ahead and like irradiate whatever crop seeds it is to try to hope for beneficial mutations, right? So you can't respond as fast. You got to wait for the new alleles. You certainly can't respond when your immediate response is going to be a lot smaller.
But the example you gave is in an artificially controlled environment where basically the ones that are not selected for technically still won't die per se. They just, humans will allow for them to survive until a better mutation comes along. Whereas in the wild, perhaps they can't evolve because they don't have the alleles away from a certain harmful trait. It could theoretically lead to the destruction of the species.
Yes, absolutely. But you have to remember that there are gradients in fitness here, and so maybe everybody's fitness is pretty okay, right? But that these guys have a lower fitness than the medium guys. So it's not always just everybody dies or everybody is totally sterile versus the other. And it also could be a trait that has some impact on fitness, but not 100% determining fitness, right?
Yeah, notice the curve. The curve only takes out one small fraction of that tallest phenotypic peak there.
Right.
Which means most of those individuals are surviving or reproducing. It's just a small number of them.
Right. So it's not necessarily it's not totality.
It's relative.
It's relative, right? It's not total fitness. So maybe everybody's making 100 offspring, but the ones that are more fit are making 400 offspring, right? So the species could survive, but you're right, in some cases this will cause extinction. Okay, so if we don't have as much additive genetic variation, we can't have as an extreme a response, and we can't have as fast a response, typically. That is Fisher's fundamental theorem, okay? So Fisher's fundamental theorem says and this is not in the exact words that he used, because the words that he used are pretty, I don't want to say arcane, but Fisher was a really smart guy around in the very early 1900s, and his wording makes sense in the context of his paper, but would be a little bit confusing. So I'm going to say the greater the genetic variation for trait, the more effective natural selection is. She can respond more extremely and faster. In fact, this has been around, like I said, since the early 1900s. And one of the graduate students that I helped to mentor in the lab where I got my postdoc, Levi Moran, he was the first person to show experimental evidence of this. And it was a Nature paper. And I was not bitter at that at all. It's like, oh, this is the guy who I had to train that 2 mealworms will not make mealworms. He's really smart, guys. I can't begrudge him that. But I'm super envious because he got to be on Through the Wormhole with Morgan Freeman. And I told him, I said, seriously, you gotta make that like your answering voicemail message. Like, you have reached Levi Moran. Right? Wouldn't you do that? I would totally do that. So this is Fisher's fundamental theorem. So if we don't have a lot of genetic variation, we can't really respond very well. It almost doesn't matter how strong the selection is. It only matters. You guys look at my shoes. Oh, there's not. It's brighter. Here you go. What's that
It's just a little smack.
Where'd he go? Oh, there he is. It's in my paper. There you go. It's like, this is not cool, man. Here. Poor little guy. All right. Okay, so I rewrote Fisher's fundamental theorem for you. It'll be on their slides. So how do we measure genetic variation? Easy. Already talked about it. Heritability. Okay, and there are a lot of different ways to measure heritability. The heritability, we talked about, okay, you've got it, if a trait is heritable, it's genetically determined, at least partially, and there's genetic variation for that trait that is reflected in the phenotype. There's actually a mathematical definition of heritability that reflects this, and it's basically, and heritability is indicated by a little h squared. It's not actually squared. That's very confusing.
Don't take the square root out.
Do not take the square root of it. It's just stupidly h squared.
They could have just as easily said heritability is Q.
Yeah, no, I don't.
It's the same thing.
Yeah, it was kind of a dumb thing. But so heritability is equal to the genetic variant, additive genetic variance over the total phenotypic variance. Okay, so basically, how much of the variation is caused by genetics versus how much of the variation is just caused by the environment, how you were raised, etc. So this is phenotypic variance, and this is additive genetic variance. Okay, so it's basically the ratio of total variation that is due to genetics, particularly additive genetic variation.
And there are literally entire books written about all of the different ways in which you can estimate that.
Yes, and my first publication basically was in response to a giant argument, and I was a tiny undergrad. Well, it was my first publication. I guess it was my third publication, but the first one that I wrote all of, and it was saying, oh, these people really didn't estimate this thing very well because there are these complications in estimation, and it was this giant argument. People in quantitative genetics are super brutal and cutthroat, and they basically will be like, no, you're an idiot, and they'll publish that. And so I was terrified to present on that paper, but I did, and it was fine. Okay, so basically it's the ratio of genetic variation to total phenotypic variation, okay? One way of estimating it is simply by plotting the parent values of the trait against the offspring values of the trait. And you would think, oh my God, that's way too easy. How in the world can you just measure the phenotype and plot them against each other and get something that is heritability? And I'm telling you, you can do it. It gives you that value. That slope gives you the value. This is called a parent-offspring regression. So you take what's called the mid-parent value, which is just the average of the two parents, and then you take the average of the offspring value. So for example, with this dot that's right on the line, the parents were something like, 65.5 inches tall, average, and then if they averaged the offspring, they were just about the same. And if you do this, the slope of this line will give you the heritability, which is donkulous, right? It's like this super easy trick. This is why I did quantitative genetics for my PhD. I thought, this is stupid. You don't have to know anything about the biology. It's fantastic. Measure the phenotypes, you do math. And it's like, oh, there are this many chromosomes involved, and blah, 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 blah. Super easy.
It actually turned out to be that easy.
It's actually super hard. It's super hard, but you don't have to know anything about the molecular mechanisms underlying them. The ironic thing is that that's what I've done with the entire rest of my career, look at the molecular mechanisms underlying them. Okay, so but no, remember, I'm super mean, right? And I don't like short people. And so I'm going to perform artificial selection on humans, and I'm going to say, okay, well, here's the average height, and I'm going to say, anybody below average, you ain't going to breed. I'm going to be super mean. And you know what? Now I've got my value of h squared. I've got my heritability value, and in this case, that slope is 0.42, right?
0.92.
0.92. Sorry, I was looking at the wrong number there.
So, and that's a ridiculous slope for high infant.
It's silly.
Lower than that.
Yeah. Anything close to 1 is probably not very realistic.
Because This
0.92.
was one class of first-year students in Evolution.
Was it really? So these are real data.
These are actual data from students, yes.
Nice. Okay. All right. So,
Is there a equation of the graph?
pardon?
Is there a equation of the y?
I didn't quite
Is
hear
that
you.
a rule of the graph?
Is it a what of the graph?
The y bar star is open. Is that a rule of the graph?
It's the yeah. So that's describing the slope of the line and the rise over run. It's like y equals mx plus b.
And this is not a 60 which is 5.6.
Don't understand what you're asking me. I'm sorry. Do you guys know what he's asking?
Yeah.
Oh, why is the y-intercept at 60? Gotcha. Well, I guess there were no.
Y-intercept is the y-intercept for 0, not 60. That's why the y-intercept is 5.
Yeah. So if you look here, this actually is 57 and 60, just because there weren't any mid-parent heights that were less than 60. But there were some offspring heights that were less than 60, and so this is kind of zoomed in on that portion of the graph. Okay, now I can understand what you're asking.
And the red line is the 1-to-1, the slope of 1.
Yeah, so red is slope of 1, and then the dashed one is the actual regression itself, which is where we get our value of here. So if I eliminate all of those individuals within that square because they are too short for me, then I'm going to get a response to selection in following generations where the height is going to increase, right? Assuming that this is a genetically based trait. And the response to selection can be thought of as R, and you can actually do this as an equation, okay? So you can calculate exactly what the response is going to be. Okay, so I've eliminated all these short people, right? And now I have a new mean height for the population. It's more closer to 70. So this equation is the breeder's equation. And you get R, which is the response to selection. Sometimes you see it as a delta Z. Just depends on who's talking to you and how they define it. It doesn't really matter. It's the same thing. So R is the response to selection. You already know H squared. That's heritability. S is the strength of selection. So in my research, I do artificial selection on these little soil worms, and I have made them super resistant to hydrogen peroxide by submerging them in hydrogen peroxide for 4 hours at a time. Initially, 90% of them died when I would do that. You might not be surprised to hear that. And in that case, my strength of selection was 0.9. 90% of them were dying. I mean, basically, 90% of them had zero fitness because it was killing them. Now they don't really care that I'm submerging them in hydrogen peroxide for 4 hours because they've responded very dramatically because their heritability for this trait is very high. Okay? So this response is the heritability times the strength of selection. If you just select on something just a little bit, like I'm going to go up and be like, hey, Austin, you know, and slap him in the arm, that's not going to impact his fitness very much, right? And so his response to my quote-unquote selection is not going to be very great. However, if I murder everyone who is short, that's going to be a response to selection. That's strong selection, right? Okay, so. Or murdering people who walk off with micropipettes. Last Thursday, I thought I was going to cut a bitch. It was not okay. Somebody was like, how come you broke a pipette? And I was like, if I had one. So mad. I'm telling you, me and Dr. Wetmore, you do not want to cross us. Woo! All right, so if we take our data from that graph that we had, okay, so we had our heritability was actually, you know, the rounded version was 0.92, it's actually 0.9165. Here's our strength of selection, 2.41. And so our response is going to, and this is actually in inches. This is actually in the units of the trait. Okay. So our response, and in my case with the worms, the trait I was interested in was survival. And that's why it was, strength of selection was 90% because I was killing 90% of them. In this case, we're getting rid of those that have height 2.41 inches below our acceptable minimum. Okay, and our response to selection is going to be an increase in the mean height by 2.209. All right, so then you can continue to, or another way to calculate this is to, your response to selection is the average offspring height after the selection minus the average offspring height before the selection. So basically you're just saying, well, what's the difference in the average after I selected? Right? It's just the response to selection. But the cool thing is that you can absolutely predict it in this way. So then I have this other sort of distribution here. Do I have as much variation now, though, as I did before? No. If I want to continue selection, what does this reduced variation mean for my success in future selection attempts? It's going to have less of an effect. Why? Okay. Why is heritability less? Hold your tongue just for a little bit. Photosynthesizers. Why do I have less heritability if I still have all these guys? Chris?
You're going to lose heritability because you're losing genetic variation.
You're losing genetic variation. Absolutely. Yeah. So you're losing genetic variation. If you think back to this heritability question, you're losing some phenotypic variation as well, but you're losing a lot of this phenotypic variation, the variation induced by the environment, should remain the same. so this is total phenotypic variation. This is genotype, environment, maternal effects, everything, okay? So those other forms of variation are gonna stay about the same, but your genetic variation is gonna go down. And so that ratio is gonna change. And so then is my selection gonna produce as large a response? It can't, right? Because R equals H squared times S, okay, but H squared is lower now. So my R has to be lower. And indeed, you can see this in the finches that were studied on the Galapagos Islands, okay? So in 1976, when they went and measured the build depth, here are the mid-parent values of build depth. So this is like average mom and dad values. Offspring build depth, average all the offspring per mom and dad, okay? And so they were plotted here, right? Then there was a weather event. There was a weather event that changed what seeds might be growing on the islands. And when they came back, they plotted it again in the blue. What do you notice about the blue compared to the red? Either the dots or the slope of that line. Bryce, you look like you want to say something.
Right, the ones from 1978 have less drastic outliers.
Okay, the ones from 1978 are clustered a little bit closer together. Yeah. Maybe less variation. Okay, what else do you notice?
Ones from 1978 just tend to be bigger.
Tend to be bigger as a whole? Yeah, absolutely. Okay, I'd agree with that. What do you notice about the slopes of those two lines? Anna, what do you think?
They're both positive.
You think one is sort of more sloped than the other? One is more flat than the other, maybe? Okay, which one looks more flat? The blue. Okay, and so the slope of the line is equivalent to which portion of our Breeder's equation? The Breeder's equation is the response equals the heritability times the force of selection. What the slope is equal to which part here? Not R, not R. It's equal to heritability, right? Because that's one way of measuring heritability is with the parent-offspring regression. Yeah. So we have less slope on this regression line. That means lower heritability. And that matches with what you guys observed, right? That the blue dots are clustered more closely together, and in general, they're larger. What do you think that the weather event did to seed size on the island? Increased it. So what do you suppose? Yeah, so we've got now this selection on peak size this way. We've lowered our heritability a bit. We've increased the mean for build depth in the population. And we've decreased the total range, right, of sizes. Questions? All right.
Yeah, just to add one thing.
Yeah.
The parents in the new generation are essentially offspring from the red generation. So the red parents gave rise to red offspring later. Essentially, the parents of the blue generation are the children of the red generation. So this is a multi-generational data set.
Yeah, that is kind of nice because when we're talking about how things change over time, really what we mean is how things are changing over generations. And in this case, the birds will breed at least once per year. Do the finches breed more than once?
I think they have actually breeding season. Usually it's
Yeah, so they have like one clutch probably.
Yeah.
Pretty common for birds.
So as time goes on and there's like greater selection in it, like for this clearly S was pretty big-ish to cause that shift. Well, does the result of a big R from one generation result in a smaller A squared for the next one?
Yes. Any R will basically result in a smaller A squared for the next generation.
Ironically, having a large heritability at first will result in much less heritability later.
If you have strong selection, yeah. So, and then that's assuming that you don't have an influx of any nutrient variation, which you could. Yeah. All right. So that's what I have, and I'm sorry that it took so long.
Before you leave, do you need to work one student in groups based
I
on
do, absolutely.
Just understanding that.
We had an insightful comment from Miss Katie Butod here in the, what group are you guys? Tenacious Turtles. We had Bryce here in For Fast for Curious. We had Connor willing to take a risk over here in Chicken Strips.
All right.
And Hannah over here in Photosynthesizers. Am I forgetting anybody?
Isaac, I feel like I've heard from him all day.
Well, we've heard from Isaac a lot, but I think we also heard from Taylor right at the very beginning. And so one for that group.
Right.
Yeah. All right. Okay, so do you want to talk about anything about your topics before the end of class, or do we not have enough time?
Yeah, just give me about, I don't know, 5 minutes.
Okay, yeah, that
Just
sounds good.
so-
Because this is all I have. I'll post this and everything that we talked about on Friday on iTunes. You should be able to do it pretty soon after class.
Yeah, so I'll just go ahead and introduce these.
Yeah, sounds good.
We're about a day behind, so if you're keeping track of how I get to your Dropbox, is it findable in your planner? So we're about a day behind. So we were supposed to cover density-dependent population growth today, and then cover interspecific competition on Wednesday, and then cover adaptive radiation on Friday. So each of those days has some homework associated with it. So in the schedule, you're going to see that there's an adaptive radiation homework assignment. We're just going to nix that, because of all the things we need to talk about before you get to class on Monday and are going to be discussing the Henry et al. Paper. One thing that you need to know least about is adaptive radiation. And the reason that we spend a day on it is that in the past, students in this class have had trouble dealing with what an adaptive radiation is and understanding how adaptive radiations occur. But I'll just try and fold that into what we do on Friday. There are reading assignments though, so you definitely should read the section in your textbook that deals with adaptive radiations, because the Henry et al. Paper deals with how adaptive radiations occur and why adaptive radiations can be reversed in a sense. And so while we're not going to spend a whole day on it, we will talk about it on Friday. So I'll send out some preparation homework for you for Wednesday, and then the homework that is in the daily schedule that is for Wednesday is probably going to be for Friday. So I just want to kind of give you a little bit of an introduction, so when you get that, that homework, you have an idea of where we're going with it. So we've been talking about natural selection, and these are just to remind you of the preconditions. And Dr. Reynolds has been talking to you mainly about these three right here. talking about where heritable variation comes from, talking about how you get from genes to phenotype, and to a certain degree with genetic selection, what phenotypes have to do with whether or not you survive and reproduce. And so that's pretty much what she's been taking you through. So if you're trying to put that information somewhere, that's all in here. What I'm going to talk to you about today and then on Wednesday and into Friday is the first one of these, which is this, what Darwin referred to as the struggle for existence. And this comes about when organisms are pressed for whatever reason to survive. They have more difficulty surviving and reproducing than perhaps they could under different conditions. And so we generally think of this as being the result of the fact that organisms can produce lots of offspring. I submit to you the dumpers. Human beings can produce lots of offspring. And only some of those, if resources are limited, are going to be able to survive and reproduce. So when we talk about this, oftentimes one of the ecological interactions that gets invoked in causing the struggle for existence is competition. And you can have competition for all sorts of things. What are the kinds of things you can compete for? Food? Who said food? Yeah, okay, Gabe. What else?
Yes.
You can compete for space. Someone else.
Mates.
Oh, you can certainly compete for mates. All I can tell you is go to a bar. And
Don't go to bar yet.
if you're old enough, you can go to the bar and you can demonstrate this. You can observe this for yourself on a nightly basis. So all organisms are competing for things, and these things that you're competing for affect either your survival or they affect your reproductive success. And because competition can affect either or both of those at the same time, ecologists have really focused on competition a lot, to the point that in the 1980s, they had concentrated on competition so much that there were another group of ecologists who said, stop concentrating on competition, you're forgetting all of this other stuff that can also happen. But in this case, because the beaks of the finches are all about getting food, and food is one of those things that we can compete for, the case study that we're doing with you guys does focus on competition, so we're going to talk about competition. It isn't the only thing that happens, it's just one of a variety of things that can happen. It just happens to be the thing that happens in this case. So, having trouble finding enough resources, it affects your growth and survival. It affects the amount of energy that you have to allocate towards reproduction. So maybe you find a mate, but because there's food limitation, your mate can't convert that copulation into producing viable offspring. Well, just the act of mating alone doesn't get you there. You actually have to produce offspring. And you have to, of course, find a mate. So we have two forms of competition. One form of competition is competition within a species, intraspecific competition, and one form of competition is competition between species. And we'll talk about both, but we're going to begin talking about intraspecific competition. And that occurs when you have individuals of a single species competing. So what do you think the main consequence of intraspecific competition is? Somebody, Chris, do you have an idea? Is that your hand going up?
Yeah.
Sorry, Isaac, I know you have an answer, but you've talked a lot today, so I'm moving to that.
Could the consequence possibly be like your species like declining if there's too much?
Okay, so if your species is declining, let's not think about species, but think about populations of individuals. What's happening to populations? They're declining. So what's being affected?
Like variation be affected by that?
Not variation isn't what I'm thinking of. If your population is declining, what's happening? Think of vital rates that you talked about in the past. Talked about this on the first test material. Mason.
There's a decrease in reproduction. There's a decrease in reproduction.
Okay, so there's a decrease in reproduction. So if you have intraspecific competition, one of the things that could be happening is the birth rate, the per capita birth rate might be going down. What else might be leading to a declining population? Now we're on this track. Let's finish up. What else would be happening? birth rate can be declining, what else can be happening? Price.
I was gonna say that rate is increasing.
Death rate can be increasing. Give a point to that group back there. So the consequences of this, what?
Price per view, but.
Oh, I'm sorry, I didn't
Okay,
hear
you just
that.
said it really quiet.
Okay, one point taken away from the stealing. No, I'm joking. And a point to Chris's group for taking a chance. So when you have intraspecific competition, because you're talking about the same species, the effects of this in terms of population growth, which is what we're going to be talking about at length on Wednesday, is you just have lower levels of population growth. Well, remember back when we talked about population growth, that's determined by how many offspring you're producing and how much death is going on. And so we're going to be talking about the effects of resource limitation on birth rates and death rates. So these are the learning objectives for the next thing that we're going to be talking about on Wednesday. I'm not going to take the time to go through them. Do we need to get anything else back, or did you get everything back that needs to be handed back?
I handed back everything I have. Someone asked about the comments on the introduction section. I do have those, most of them. You can come by my office to get them. I thought I had them in my bag, but I don't. You can come by my office to get them, or I'll give them to you on Wednesday.
Okay, so I'm not going to go through these, read them. We'll start here on Wednesday. I am going to give you a preparation homework assignment. It's not posted yet, but it'll be posted later on today. I just didn't know whether I needed to post it. So be ready for that.
Oh, wait, before you go, please bring your test corrections.
Oh, yeah.
Those are due today.
Test correction. Are you finding the test correction thing useful? Okay, all right, good.
Yeah.
I think, but it should have a due date on the assignment, and I think it's dated. Which for you guys are getting last.
Yeah.
Yeah, you should get a point, not us.
Huh?
I don't think you get a point to us.
Well, so one of the things that we want to encourage with this is we want you to be participatory, and just because you're not always right doesn't mean that you don't get credit for participating, because we would rather have you participate and occasionally be wrong than not participate at all because you fear. One of the things we find from students coming from high school is you fear being wrong. Well, okay, fine. So we want to create an environment where you can take chances and be wrong and still get credit for participating. There are going to be times when we won't give you points because we really are looking for the correct answer. But like what we were just doing, I'm asking you to reach back and recall stuff from first test material. I don't necessarily expect you to be correct in that case. Yeah. So don't sweat it.
Yeah.